Now, I'm pleased to introduce our second speaker, Dr. Ying Li. Dr. Li is the Director of Biologics PD Department of ProBio, where she leads CMC process development and platform innovation. She has successfully led the development of over 20 CMC programs, supporting R&D approvals across the FDA, NMPA, and EMA. Prior to joining ProBio, Dr. Li served as R&D manager at Merck KGaA, where she focused on developing platform processes for biologics and viral vectors. She holds a BS and PhD in chemical engineering from the National University of Singapore and the Pennsylvania State University, respectively. She is a frequent speaker at international conferences and has co-authored 9 publications and 4 patents in the field of bioprocessing. In this talk, Dr. Li will discuss from VHH to LNP, antibody fragment process development empowering precise targeted delivery. Now I will hand it over to Dr. Li.
Thank you very much, Jessica, for your kind introduction. And hello, everyone. Welcome to join today's talk. All right, so here is the contents to be covered in my session. We'll start with a general background information of antibody LNP, and followed by our general CMC strategy and case studies for different types of antibody fragments. So antibody fragments have been increasingly popular in use of antibody engineering, as well as a number of different conjugation applications. So there are a number of key advantages of utilizing antibody fragments, including their small molecular weight with strong tissue penetration, which enables improved permeability through solid tumors and tissue areas. Their simple structure with flat copy design is ideal for antibody engineering, which easily modified via genetic engineering to construct bispecific, multispecific, as well as other advanced formats. And we have seen these antibody fragments being used in a number of different conjugation applications, including but not limited to TLNP, but also radioisotope antibody conjugates, as well as antibody oligonucleotide conjugates, etc. Their simple formats facilitate efficient conjugation, as well as minimize Fc-related immune activation of targeting binding. So here is an overview specifically to antibody TLNP applications. So I think there's also relevant questions just raised up from the audience. How is typically antibody conjugated to LNP? So there are generally 3 different approaches. One is called one-pot synthesis, where we can mix everything, all the components in the one pot and formulate the TLNP in the one-stop solution. And the second approach is generally formulate the LNP mRNA first and conjugate the antibody or antibody fragments on the surface of the TLNP. And this is generally the most commonly used approach for the majority of the platform. And the third way is called post-insertion. Basically, you can conjugate the antibody to the lipid PEG molecule and then formulate the TLNP in the second step. And just to note that for today's talk, we are only going to cover the CMC program for antibody fragment, but not for the full stop solution for mRNA and TLNP CMC. And just to know that ProBio can offer a one-stop solution from antibody to mRNA to TLNP formulation. And I think this could be a topic of future interest. And so in terms of the different type of antibody fragments, we are going to touch base on the CMC strategies for 3 different formats, including single-chain FA, VHH, as well as Fabs. So in terms of the expression system, there are generally two different options. One is the CHO-based cell expression, and the other one is the E. coli-based expression.
Next, let's move on to our CMC strategy for 3 different types of antibody fragments, starting with Fab fragment. There are a number of challenges associated with the Fab process development, including low expression titer, imbalanced chain expression, and limited protein stability, especially prone to form high level of aggregates. So as a platform, we developed optimal solution, including utilizing a dual vector design and optimize the vector ratio to minimize the misbalanced chain expression, as well as optimize the expression level. The clone screening will be conducted to select the high assembly efficiency and optimize the feed strategy to improve the expression titer. For the purification part, since there's no Fc structure and high aggregates and potential homodimer and fragments will also be present in the cell culture, we developed a platform solution targeting specifically to address these challenges for single-chain FV, which will be shared in the next couple of slides. And in terms of the homodimer, both covalent and non-covalent species will be present. For the analytical challenges, we developed our analytical platform method and validated for challenging impurity qualification.
Let's restart from this slide. So in general, for this Fab molecule, utilizing our platform cell line process and upstream process, we yield a pretty decent titer, which around 8 grams per liter. So the main challenge lies in the downstream to identify a good capture strategy for this Fab molecule. So in general, we adopted a comprehensive screening for different types of resins, covering different domains for the Fab, including VL, VH, CL, and CH1 domains. And as you can see, actually, the different domains based affinity resin offers a very different binding capacity. And as a result, the VL domain, certain capture resin yielded much higher capacity and in general result in a significant reduction in the overall manufacturing cost. And in addition, we developed the process to effectively remove the aggregates as well as a homodimer present, which is a light chain homodimer. So through this affinity step, we initially started with a linear-based gradient elution and identified that at proper conditions, we can have a very good separation between our target molecule, the light chain homodimer, as well as the rest of the aggregates. So based on this result, we can convert that into isocratic elution process to effectively release our target molecule with pretty high overall SEC and CE purity, as shown in this slide. As a result, for the overall process for 3-step chromatography, we can yield a pretty high purity as well as low residue impurity levels. And overall recovery is greater than 70%. And here discuss a couple of analytical challenges we explored through this project. So first, for these applications, we identified that actually the ELISA potency as well as the toxicology increase significantly as with the level of SEC aggregates impurity. So as shown in this table, with a slight increase of the aggregates level to be less than around 1%, we can see a significant increase in the ELISA potency, as well as the increased level of toxicology. Therefore, in terms of the CMC risk, we have to control the overall aggregates level to be minimal at least less than 1%. So luckily, this demonstrates the robustness of the process we developed through several GMP production and scale up. We successfully demonstrate a controlled level of SEC purity to be above 99% for all batches delivered. On the other side, we did a deep dive on the light chain homodimer species present in the impurities. As you can see from the CE-SDS-NR profile, the majority of the impurities actually is missing the disulfide bond from the two light chain species, indicating that this assemblies through the hydrophobic or non-specific interactions dominate the impurity species. The next is the single-chain FV fragment, and as Derek mentioned in his talk, this could be one of the most challenging antibody fragments in terms of the CMC development and developability. So in general, we find lower expression level compared with other antibody and antibody formats for single-chain FV, much higher tendency to form aggregates. In general, even more than 20–30% is not uncommon for single-chain FV species. Very limited protein stability and prone to form precipitation and aggregates as well, and sometimes result in lower cell viability during expression process. And so the design of the single-chain FV plays a key role in their developability. In our platform experience, the design of the interchain disulfide bond, the design of the interchain linker, as well as even the order of the single-chain FV and the way to connect with the other antibody domains all plays a key, has certain impact on the overall stability of the antibody design. And in terms of the downstream, similarly, we have to design a specific capture step to maximize the binding capacity and also the purity as well, and how to control the aggregates and maintain the stability of the fragment is also the key for the success for the CMC development. So for one of the studies for the CMC program we have delivered in the past, we identified that the temperature drop time and the set point is critical to maximize the expression level, as shown in this slide. So comparing the temperature shift at 37 degrees, 33 degrees, and 30 degrees at relatively lower temperature, it helps to better maintain the stability of the cells. And as a result, the titer and QP of the cells increase significantly at relatively lower temperature. So as a result, we systematically optimize the upstream platform process for this molecule, and then successfully extended the cell culture duration from 15 days to 17 days, and improve the titer of expression by more than 6 times. Moving on to the overall downstream process, this is a rather complicated case since we have to construct 2 different single-chain FV species and conjugate a linker to the C terminal of each of the single-chain FV. And afterwards, we will conjugate the 2 single-chain FV species through a 3rd step conjugation. So in the overall program, we designed the capture process, as we mentioned, based on the protein L-based process for both single-chain FV, as well as optimizing the conjugation process for each molecules. And afterwards, specific polishing steps to remove the majority of the aggregates, and as well as the post-conjugation polishing steps. As you can see, this is rather uncommon for the quality data for single-chain FV species. So initially, from the cell culture process, the purity is rather low. The main peak only count for less than 50% of the overall product. So effective capture step and polishing step have to be developed to remove the majority of the impurities present in the supernatant. All right, so the next slide is also related to the design of the single-chain FV, its stability. So in this case, the sequence of the VH and VL significantly impact the quality and titer of expression. So as shown in here, after alternating the design of the single-chain FV, we have observed a significant increase in the titer as well as the purity from the upstream process. Similarly, this is also for bispecific case, but rather very representative as well. So initially, as a mild single-chain FV design, as you can see, the developability is rather poor since through a concentration to less than 10 microgram per ml, the high molecular weight species is significantly increased. And this challenge cannot be addressed through the formulation screening, or very challenging to be removed in the downstream as well. So in the early phase, pre-CMC program, we worked together with the client to design the interchain disulfide bond and successfully address this aggregates issue in the pre-CMC stage. As you can see, after modification, it helps to stabilize the design of the single-chain FV, minimize the exposure of hydrophobic patches and formation of high molecular weight species, and successfully move this program into CMC stage. So the next type is the VHH or nanobody, and in our experience, can you hear me talking now? I think I should be okay now. But so for VHH, in our platform experience, it's the most commonly used antibody fragment in the TLNP applications, given its higher flexibility and also a very good efficiency in targeting. To compare the VHH species with the human VH, it actually offers a better developability and higher stability. So the main difference between the VHH versus VH, first, the VHH has 4 different modifications at the FR2 domain, which replace the original VH 4 hydrophobic amino acids with 4 hydrophilic amino acids, and which increase its solubility. And in addition, there is going to be interchain disulfide bond between the CDR1 and CDR3 regions, which further enhances the structure stability of VHH. So in our experience, the VHH has very good developability in terms of expression level as well as the purification process. So in our experience, there are 3 general different types of VHH being utilized in the TLNP applications and other applications as well. So first is the most commonly used is the VHH with unpaired cysteines in their C terminals, which enables very easy conjugation through the next one-step conjugation to the TLNP surface. And alternatively, some people design oligopeptides on the C terminals, which also enables a site-specific conjugation in the next step. For other cases, for VHH, not for conjugation purposes, we have the VHH nitro format connected in tandem. So the key challenge for the VHH especially for some VHH connected in series. And similarly, since there's no FC structures, people normally could adopt protein A-based capture strategy to capture the VH3 group. However, for certain VHH families, which lack of VH3 families, it cannot be captured by a typical protein A-based affinity resin, such as PRISM A. So we have to develop alternative solutions to capture the VH. And in this approach, we have to consider the HCP removal if the affinity-based chromatography is not being used. And next is if the unpaired cysteine is present in the majority of the cases, the dimer, VHH dimer will be present and should be considered in the downstream process. So let's move on to the first type of VHH, which is naked VHH. This is a rather straightforward case, and utilizing this VHH has the affinity for protein A, which belongs to the VH3 group. And so the main challenge is rather low capacity if we utilize a PRISM A to capture the VH3. Generally, it's only could take for like 10 to 20 mg per ml as a capacity, which could be lower than the typical IgG programs. And in addition, given its smaller size, molecular weight, we have to design specifically the UF-DF step utilizing small molecular weight cutoff to prevent product loss. So in the majority of the single VHH cases, we can adopt two-step chromatography, first based on PRISM A capture and followed by typically AEX step to effectively remove the majority of the impurities and maintain a decent level of residues for next step conjugation, for the next step applications. And for the second case is a VHH with a C-terminal unpaired cysteines. The expression level is also pretty good with one round of optimization already reached 6.5 gram per liter. So one of the questions that might be raised is also pretty common, since there is a presence of unpaired cysteines, which is going to be used for next step conjugation to the LNP. However, during the CMC stage for VHH, the VHH dimer could be present due to the disulfide bond formation. To deal with this, there are two different approaches. So the first one is we can treat the dimer as our target product, since given the next step is the reduction step to enable the further conjugation of cysteine with the LNP surface, all the PEG lipid, we can treat this dimer as our target product and move forward. And this could be the choice for a lot of clients. Alternatively, if you would like to have this being converted to the monomer species, we can add in some even mild reductants, such as cysteine in the affinity alloys, and keep that in the process. We can successfully maintain all the species into the monomer format and move on to the next stage. So as a capture step, as I mentioned before, there are generally two approaches. First is to use affinity-based approach for capture, typically PRISM A-based affinity resin if it has VH3 group. And the advantage is that it has a pretty low residual level, such as HCP. However, it has rather low binding capacity, and also it will capture both the monomer and dimer, and typically they are difficult to be separated from each other. An alternative approach is to utilize other mode of chromatography, such as CEX or mixed mode CEX, to capture the VHH. So so ion exchange could basically achieve a better separation of the monomer versus the dimer, and in general offers higher capacity. However, the HCP level could be higher. Therefore, generally, that requires a three-step chromatography to effectively remove the rest of the HCP, HCD, and other residual levels if we utilize the mixed mode approach. All right, so the third group is some VHH, different type of VHH species without the protein A affinity. So in general, we can adopt the mixed mode CEX or CEX as a capture step. The initial purity in terms of ICC could be very high, and it is generally the case for VHH molecules. The advantage of using mixed mode CEX is that typically it could load at rather higher salt concentration, so there's no significant dilution of the pool needed at the beginning of the harvest process. So here is one of the case for the VHH in tandem. This format sometimes has issues with dropped viability level as we observed in the initial stage of development. And basically we identify that, again, the temperature shift plays a key role in maintaining the viability of the process. As you can see, both the temperature shift point, in particular from day 5 to later date, has a significant impact on maintaining the viability level. So basically extending the temperature drop time could help better maintain the viability of the cell culture. And also, if this is the case, a careful screening of different combination of media and clone could help to further improve the viability as well as the expression level. So VHH in tandem, unlike the VHH alone, sometimes has some stability issue. In particular, sometimes at certain salt and pH combination, it could form precipitation. And low pH, under low pH condition is not very stable, which leads to the formation of some aggregates. And in addition, if we use the mixed mode CEX as capture, we have to design effective step for HCP removal. And generally it could be addressed in the follow-up for polishing step, such as adding additional wash step and cutting the fractions of elutions could help to minimize the presence of HCP level. And last but not least, to maintain the stability of the molecule, we find for this molecule in particular, adding some metal ion could help to effectively prevent the formation of aggregates. As you can see, before the optimization, during nanofiltration step, there is a very rapid decline in the flux. And after adding certain metal ion as additives, it's effectively helped to stabilize the protein and maintain a high throughput during the nanofiltration process. And here is a summary of the overall process for this VHH in tandem case. So in summary, as a platform, we covered the CMC programs for the antibody part, in particular antibody fragments for TLNP and other type of conjugates applications. So as a platform, we offer a one-stop solution from early phase developability study to process development, as well as the follow-up, the conjugation process development, formulation development, analytical development, as well as large-scale GMP manufacturing. The overall timeline, depending on the client's requirement, the general platform offering could be 4 to 5 months from the DNA synthesis to the tox batch release. This is based on the PCB single clone production. And depending on the different requirements, we have seen a number of different requirements for this antibody, like projects such as for IND purposes, for IIT purposes, and the majority of the cases is considered not for therapeutic use, but rather for the API. So we have developed tailored CMC programs with shorter development overall timeline to meet these specific needs. With that, that concludes the contents for my talk today. And thank you very much for tuning in. And let's see if there are any questions to be addressed. Thank you.
Hi, Dr. Li. Thank you for your presentation. Since we have only 8 minutes left, you can choose 5 questions or 3 or 4 questions to answer. Yeah, maybe I'll leave you 5 minutes for the Q&A.
All right. Thank you very much. I saw there are quite a number of questions, I think most relevant to the mRNA LNP platform rather than the antibody CMC platform. So I think one of the questions that's already been addressed is, was antibody conjugate after the LNP formulation? Yes, in our platform, process is typically, which refers to the surface conjugation case, which is formulate the LNP mRNA first and conjugate the antibody in the next step. And another question, provides surface such as blah, blah, blah. In that case, would the sponsor typically outsource this work? Yes, I think this question is better to be addressed by our mRNA LNP platform team, but to my understanding, this is generally the case. But as a platform, we can also work together with a client to help formulate their mRNA LNP if there's no platform solution available from the client.